Welcome to the Explainer. So today, we're not just looking at the human body as some biological machine. We're going to look at it as this vast, highly secure communication network. Every single second, billions of chemical messages are sent, received, and decoded inside of you, keeping you alive and functioning. But what happens when we figure out how to intercept, rewrite, or even block those messages? Let's start macroscopic. Imagine an endocrine cell releasing a hormone. That's our chemical signal directly into the bloodstream. It travels like a secure message on a massive highway, rushing past countless cells until it finds the exact receptor waiting for it on a highly specific target cell. But how does the target cell actually read that message? Well, that brings us to a foundational concept: cell signal transduction. Basically, it's the translation of extracellular chemical signals into intracellular responses. In plain English, it's how a cell takes an outside message, like that hormone we just talked about, and translates it into an internal action. Now, you might be wondering, why does this matter so much to modern medicine? Well, get ready for this number: 50%. A staggering 50% of all known drug targets rely on hacking these exact cellular translation mechanisms. Literally half of your medicine cabinet is designed specifically to manipulate how your cells talk to each other. Crazy, right? So here's our roadmap for this explainer. First, we'll cover the cellular communication network, then translating external signals, followed by ligand-gated ion channels, and finally G-protein-coupled receptors. Section 1, the cellular communication network, how our cells talk. Okay, so we just talked about that macroscopic bloodstream highway, but cellular communication varies wildly in scale. Take synaptic signaling, for example. It's a highly localized pathway. Instead of a hormone traveling across your entire body, a neuron releases chemical signals across a microscopic gap to instantly trigger the adjacent cell. It's incredibly rapid, highly precise, and extremely localized. But here's the thing. Whether it's a long-distance hormone journey or a lightning-fast synaptic firing, normal physiology always follows this fundamental three-part sequence. You have a signal, which binds to a receptor, which then triggers a response. The absolutely crucial point here is that without a receptor to catch the signal, that chemical message is completely useless. The receptor really is the hub of all the action. Moving on to Section 2. Translating external signals, the 4 receptor classes. So, there are 4 main classes of receptors acting as complex translators for the cell. We've got ligand-gated ion channels, G-protein-coupled receptors, or GPCRs for short, enzyme-linked receptors, and nuclear receptors. Now, for the rest of this explainer, we're going to focus heavily on the first 2, mostly because they represent massive targets for both everyday physiology and pharmacological hacking. Section 3. Ligand-gated ion channels, the fast switches. Let's zoom in on these fast switches, structurally known as ionotropic receptors. Imagine a neuromuscular junction. A neurotransmitter called acetylcholine is acting as an agonist. And what's an agonist? It's basically a signal that naturally binds to a receptor and actively turns it on. When acetylcholine binds, it physically flips the switch, opening a rapid gateway right in the cell membrane. Sodium ions, that's the Na+, rush in to create an electrical action potential. This is literally how your brain tells your muscles to move, all happening in milliseconds. But what happens when we want to, hack this system? Well, this is where pharmacology steps in. And the vocabulary here is vital to understand. An agonist turns the switch on, just like our natural acetylcholine. An antagonist, on the other hand, actively blocks the switch. Think of it like jamming the lock so the correct key can literally never fit. Take tubocurare, for example. It acts as a competitive antagonist. It's physically sitting right in the exact same receptor spot that our natural acetylcholine wants to use. Because the tubocurare is just sitting there in the way, intercepting the signal, the sodium gate stays completely shut. No action potential means no muscle movement. In fact, tubocurare is the active ingredient in curare, which is a famous paralyzing agent that hacks your cellular communication network just to shut the whole thing down. All right, Section 4, G-protein-coupled receptors, or the intracellular domino effect. Now, while ion channels are relatively simple membrane switches, GPCRs are a whole different beast. There are remarkably 865 different types of these complex routers in the human body, and they operate in this really rhythmic 4-step sequence. Step 1, an agonist binds to the GPCR on the outside of the cell, causing it to physically change shape. That's a conformational change. Step 2, this shapeshift activates a heterotrimeric G protein right inside the cell membrane.
It acts like a switch by swapping out a molecule called GDP for a highly active one called GTP. Step 3. This newly activated G protein breaks away and activates a specific effector enzyme. And finally, step 4. That effector generates an intracellular second messenger. The coolest part? The initial outside signal never actually enters the cell. Instead, it creates this massive intracellular domino effect to get the message across. If that sounds like some super dense biochemistry, let's ground it in everyday reality. You are heavily relying on GPCRs right this very second. They are directly responsible for your light detection, which is allowing you to watch this explainer. They drive odorant detection, hormone detection, and the neurotransmission, keeping you focused right now. They are, for all intents and purposes, the primary biological interface between you and the outside world.
And because they're absolutely fundamental to how we experience the world, they are the ultimate target for pharmacological system hacks. Let's bring this concept back to your medicine cabinet. Salbutamol, the life-saving asthma inhaler, targets the beta-2 adrenergic GPCR to rapidly open up your airways. Morphine hacks the mu-opioid receptor to kill pain. Loratadine, your standard over-the-counter allergy pill, blocks the H1 histamine receptor. Wildly different drugs, wildly different physiological effects, but all of them are hacking the exact same GPCR domino mechanism. Section 5. The key to targeted hacking, molecular variation. So how in the world can one GPCR system do so many wildly different things without constantly crossing wires? Well, the secret is elegantly simple. It's molecular variation in the alpha subunit of the G protein itself. Because there are different types of these subunits, like different molecular flavors, if you will, each specific receptor can couple to a very distinct, isolated intracellular signaling pathway. It's the exact reason an asthma inhaler opens your lungs but doesn't accidentally block your pain receptors. And this leaves us looking directly at the true frontier of medicine. With molecular variations like the alpha subunit providing all these distinct, highly specific pathways, what untapped cellular conversations could the next generation of drugs hack into? I mean, if we already leverage 50% of these cellular targets to treat the illnesses of today, just imagine the diseases we could cure when we finally decode the rest. Keep that in mind the next time you take a simple allergy pill. Thanks for joining me on this explainer. Keep learning, and we'll see you next time.